I'm going to be introducing Olga Babinek. She's a genomics expert applying computation to biology, and she currently works as a bioinformatician at the Chan Zuckerberg BioHub in San Mateo, where she studies grand unified theory of cells by applying computer science and machine learning algorithms to biological data. She has two BS degrees from MIT, one in mathematics and one in biological engineering. But I promise you can ask her questions and she'll give you answers that you understand, which she says she's open to. She has a master's in bioinformatics and biological engineering from UC Santa Cruz. And finally, her education was completed through getting a PhD in bioinformatics and systems biology at UC San Diego. She's a NUM-focused John Hunter Fellowship and NDSEG Fellowship recipient. And she runs a Twitch live stream channel called Bioinformatics Beyonce, which I suggest all of you guys go and check out. And today, Olga is going to talk about single-cell RNA-seq. So will you please welcome Olga Babinek.
Can I ask before we start, who here has heard of single-cell RNA sequencing? Who here has worked with or been around people working with single-cell RNA data? Who here is sitting on a pile of single-cell RNA data that you're going to be able to do? Okay, cool. Just out of curiosity. And so to give you a brief overview of what we'll all talk about today, I'll give a brief introduction about why maybe you've heard of single-cell RNA-seq, but why you should care about single-cell RNA sequencing. I'll give an overview about best practices in single-cell RNA-seq analyses pipelines. These slides are largely from my collaborator, colleague, Angela Pisco, so huge thanks to her for letting me borrow these slides. And this is, I would say, like best practices as it is now. What my research focuses on is, as Shannon mentioned, a grand unified theory of cells. So I'd like to see compare cell types across many species, especially in a reference-free way. So that's what we'll talk about a little bit in the end. And as she mentioned, please feel free to interrupt me with any questions or clarifications you may have, because it'll just make this a lot more enjoyable for everyone. So first of all, why transcriptomics? You may know already that in multicellular organisms, every cell contains nearly the same genome, but not every gene is on or transcriptionally active in each cell. And this, by measuring the transcriptome, we can have a closer view of what genes are on in a particular cell type. So we can tell the difference from the transcriptome, difference between smooth muscle, between family neurons, adipose cells, and so on. So we can really dissect the tissues that we have. Now, why single cells? So given a heterogeneous tissue, I know I think all of you were working with DNAseq2 this morning. Who was not? Anyone not?
Yeah,
Some of the afternoon? Okay. You've been exposed to RNA sequencing, from what I understand. Am I correct? Okay, cool. That's good. Okay. And so from there, you may know that given the bulk analysis, bulk RNA input, you can get an average gene expression result from all cells and may not be able to tell the difference between the different cells that contribute to this gene expression in your tissue. With single-cell analysis, one is able to capture each individual cell and then find that each cell type has a distinct expression profile, and then be able to reveal heterogeneity of subpopulations within your tissue type. Now, what is single-cell transcriptomics? Well, first of all, we take solid tissue. As I say, you have solid tissue, you explode the tissue to get each individual cell. Then from there, you have one cell at a time. You explode each cell to get its RNA content, as you would with other RNA-seq technologies. Do reverse transcriptase and second-strand synthesis. From there, you have your amplified RNA, perform more reverse transcriptase and PCR, amplified cDNA, build your up your sequencing library, sequencing, build up your single cell expression profiles, and use these expression profiles to build your cell type identification. And we'll get into more detail about the bioinformatics steps here. So here we'll briefly talk about the experimental design, leading into the reads processing, the quality control that we need to perform on each cell, and finally, I think the most exciting is the data analysis and the interpretation, but we do need to make sure we've done each step that comes there the best we can. So let's talk about experimental design. I've heard some of you talk about experimental design already, so maybe some of this is already familiar to you, but what is your source? Is it a primary organism? Is it a cell culture? What's the biological variation that already exists in your data? If you have mice of different sexes and you process the mice of different sexes completely different ways, then you confounded your biological variability with your technical variability, which will be a problem. How long your duration of sourcing? So if you're working with, say, human samples, and the post-mortem interval could be important. Is it quite variable across your samples? And so on. You may want many biological replicates of the same organism, same animal. How many cells can you get from each one? So on. Now, tissue dissociation. I am by no means an expert in tissue dissociation. I only know that there's many different ways that they affect your dissociation. One thing that can affect you is how quickly that dissociation can be done, and whether that dissociation does affect your gene expression. There was a paper I think last year published that they claim to have found basically dissociation-specific signature in their single cells. I think it was FOSP, and that this gene was particularly active in the cells that they captured, the single cells that they captured, but when they went to do validate their findings using RNA fish or like an in situ staining in their cells, they didn't see that same gene transcriptionally active. So how you do the dissociation can affect your results down the line. That is something to keep in mind. If you are interested in a particular cell subpopulation, you may want to enrich for this cell type. That does mean that you need to already know what this cell type is. You have to have a marker already existing for it so you can pull it out. But if you have both of those criteria, you can use a method such as FACS sorting to sort particular cells, or other dead cell removals and so on to pull out particular cell types. Now, the platform has a pretty big effect on the quality and the overall results that we have from your single cell data sets. Most commonly used, I'd say, are the droplet-based methods, such as from 10x Genomics, their Chromium or GenCode system, just because it's so easy to use. Like, given an input sample, you basically just get your libraries prepped for you. More labor-intensive methods could be the tube-based after FACS, or microwave-based, microfluidics-based, which tend to encode a full transcript rather than just a 3' end of the transcript. We'll talk a little bit about that. So yeah, if you're interested in, say, alternative splicing of a full transcript, you may need a lot more reads. And two, you may want to look at the full transcript as opposed to just one file tree format. And finally, we see sequencing libraries, most typically with Illumina next generation sequencing. Though there's some pretty exciting work in the PacBio IsoSeq world, though I don't know if it's really ready for everyone to use yet. I don't think it's way too expensive compared to something like 10x, which is already pricey. Yeah, and you may want to consider how deeply you want your sample sequenced. Yeah, if you care about finding differential genes and particular cell types, you may not need to. You can get away with sequencing pretty lowly. 50K reads per cell is pretty reasonable. If you really want deep alternative splicing, you do need to sequence them like tens of millions of reads per cell, unfortunately. Any questions so far? So in terms of the platforms, we have quite a few. I really like this figure that really nicely summarizes the number of cells per paper, essentially, for single cell paper. Basically, in 2014, that's when 10x released their system, and since then, there's been a huge explosion of single cell RNA-seq data sets. And there's quite a few. So first are isolating cells manually with a mouth pipette, which is kind of crazy to me. Then multiplexing into to a few wells at a time. In 2013, there was a couple of papers published using Fluidigm C1 chip, which allows you to capture one cell at a time and do a full-length transcript prep. That's the data that I worked with before. Liquid handling robots in 2014. The nano droplets is really what changed the game for a single cell. You may have heard of DropSeq or InDrops or 10x. They all use very, very similar methods to capture cells and amplify them. The chemistry is slightly different. There's interesting work done with pico wells, so like a little grid of little wells, like a little honeycomb that you try to kind of sift your cells into and hope that they land with the bead. But allows you a lot of visibility into what's going into your experiment. And finally, in situ barcoding. So really barcoding the cells and the sequences in the sample itself, which is pretty awesome. There was a paper from CC BioHub that I was involved in. We call Tibulimerase. It was about 100,000 cells from 20 different organs in mouse. That was a sex balance study, so 3 females, 4 males, as balances, I guess it is. More recently, we published a half-million cell paper of the aging mouse cell atlas. So this is of not just a 3-month-old mouse, as was in Tibulimerase, which is an adult human, but including 30-month mice, 24-month mice. So 30-month mice are about the same as a centenarian, like a 100-year-old person. So they really looked into the differences in age and mutational load and different cell types there. So another nice paper from Valentin Svensson looked into the sensitivity of these different methods. So not just sensitivity, but also accuracy. So the gold standard here is bulk RNA-seq. So if you took the same sample and really, so this is with cell lines, so really the same sample, as similar as you can get. How accurate, how similar are the recounts compared to bulk RNA-seq, and how many genes did you get compared to bulk RNA-seq per cell? So your choice of platform really affects how close you are to bulk RNA-seq. But choice of platform includes many things, not just accuracy and sensitivity, but also cost. Right, so let's talk about reads processing and quality control. Before I move on, I want to give a shameless plug for something I've been working on, which is versioned, reproducible, community-driven best practices pipelines with in a language called Nextflow. I've been working with people around the world to, in particular for RNA-seq and single-cell RNA-seq pipelines. What I really like about Nextflow specifically is that it works very well with a variety of compute environments. So given inputs that are some from the cloud, some from your local computer, it has no problem dealing with all of those inputs and getting to the same place, and then finally outputting into the same, into the cloud or local. One of the things I really like about it is we use a mix of AWS, Amazon Web Services, and local compute. So for us, having the flexibility to do either one and to have build pipelines that are usable by our collaborators with like any of the compute engines that they use has been really huge. So if you're working on some pipeline in the future, I suggest checking this out to see if something has already been written for you and you can do a little bit less work and focus more on the question at hand. So back to single-cell RNA-seq processing. So let's think about this a little bit more. So review. So we've isolated our cells into relative droplets. And each cell is lysed, we get individual mRNA molecules, and are captured here. This is where the unique molecular identifiers, or UMIs, are added for the droplet-based methods. This allows you to see that many of these reads actually came from the same molecule, and then you can collapse your signal to say that, okay, even though I saw 100 of these reads, they all have the same UMI, so I actually only have one molecule in the beginning. So it allows you to clean up your signal from what you would have to do more difficultly if you did not have UMIs. So from there, we're cDNA is PCR amplified, fragmented, amplified again to get a library prep, and then sequence, demultiplex, and so on. What can be difficult about this is, so we have our RNA sequencing reads, and we're looking to align them to the genome. We can also assemble transcripts de novo. Typically, this is not what I see people doing with single-cell RNA sequencing, unless they're using PacBio isoseq, and that assembly problem has essentially been covered for them. From here is what we really care about for quality control. What are my notes? I just wanted to double check that we get there. Okay, we'll do it out. So one thing we care about is whether each individual cell actually succeeded or failed, like which cells are go or no go. You can assess this a number of ways: number of transcripts per cell, number of molecules captured. Generally, more is better, though it can depend on the cell type, which can be tricky. Number of transcripts that align to a reference genome, and more transcripts to a reference genome, depending on your reference genome, is better. High mitochondrial RNA may indicate that the cell is actually not so great. Could be apoptosis or some other factor at the time. And overall number of genes affected. So as you can see here, the trace protocol, so orange being drop-seq, and blue being SMART-seq C1, and yellow being SMART-seq 2, you can have a twofold difference in the number of genes being detected. And that may matter to you if you need quite a few genes to tell the difference between your cell types. If you have cells types that are quite different already, it maybe fewer genes is not as big of a problem. So let's talk a little bit about the data analysis. In general, what I see people doing with single-cell RNA-seq is they're typically taking a heterogeneous population and trying to find individual cell types, or they have a particular trajectory or developmental or after a particular stimulus that they're interested in. In this case, if you're doing a trajectory analysis, it's best to have data that's on the order of hours different from one another, and the order of months may not give you the results you're interested in. So you do need to have pretty fine-grained data here to get a great trajectory. Other things I see people doing are looking for transcriptional mechanics, so understanding transcriptional bursting, whether genes are on or off. If you're familiar with the single-cell data, you may know that if you have a particular cell cluster that you know is clustering very closely together, you see all these cells are similar, and you take a gene that you know should be on in all of these cells, you see like monochrome confetti in this cluster. That some of the cells have it, some of the cells don't, and it seems really strange, but this is another case where it's not ideal to use a single gene as a threshold to call a particular cell type for a single-cell RNA-seq. Another thing people do is do network inference or interference. Maybe they do that too. And looking for genes that are co-expressed across different modules and across different cells. One of the first things people will do, one of the first things you do with a single-cell RNA-seq is do a dimensionality reduction. You may first want to do a feature selection. So, for example, for human or mouse genome, you have about 20,000 genes, and that can be kind of a lot for dimensionality reduction algorithms when in the end, you may want a couple or a handful, like 2 to 20 or 50 dimensions in the end. So what I recommend doing is to first filter for high variance genes that are informative across your data set, ones that are changing a lot, because ones that aren't changing are going to be so helpful. And from there, typically one gets around a few thousand genes, so you reduce your count by about an order of magnitude. From there, we can then do dimensional reduction techniques that either are what I would say as interpretable, that each component has some, at least mathematical meaning, that this, for factor analysis or principal component analysis, each component is some summation of genes, like 10 of gene A and 3 of gene B and minus 10 of gene C. Or a projection-based approach. These are often called manifold learning algorithms, where instead of trying to find a line that for your data that really explains your data, you're allowing it to curve through. It's kind of like when you have a coastline and you're trying to understand this whole coastline. You can't really understand it linearly. You're allowing for the small indentations for each coastline to go through. And I should caution that these projection-based approaches should be used for visualization purposes only. Something that I see people who are new to single-cell RNA-seq do a lot is interpreting the x and y axis of their TC plot to be meaningful of some particular developmental direction or something. And unfortunately, TC is stochastic, so you can't really interpret it that way. And we'll go a little bit more into how it works. So let's talk about PCA, or principal component analysis. It's a statistical procedure to transform your set of observations, so in this case, your gene expression matrix, into sets of linearly uncorrelated variables, the principal components. I like this visual. This is interesting. This person's PhD thesis, I feel, is cited in literally every talk I see about PCA, just because this figure is so good. And it looks kind of crufty, but it's better than a lot of the other PCA figures out there. And one analogy I really like about PCA is that if you think about your data as being, your cells as being fish in a fish tank, your first PC, the goal is to shine a flashlight through the most fish at once. What direction of gene expression can explain the most variance of all of your data at once? Now, if you have a very heterogeneous data set with many different cell types, you can imagine that first component that explains all of them at once is not going to be that informative for any single one. It's going to be largely ribosomal RNA, largely housekeeping things, unless you do have a very clear trajectory. Maybe that is your data, where you have from one cell type to another. Then you may have a more clear PC1. But in general, specifically for PCA, what we're looking for is to explain the largest amount of the data, largest variance of the data in the first principal component, and then every next component, you're trying to explain as much as possible. So just trying to shine another flashlight, but now your flashlight has to be at some right angle to your original flashlight. So the PCs correspond to the eigenvectors of the covariance matrix, if that math interests you. Again, the first principal component accounts for as much of their variability as possible. So what's mathematically nice about them being the eigenvectors of the covariance matrix is that they are sorted in order. So PC1 is always most important, PC2, PC3, PC4. And what we typically do for single-cell RNA-seq is look at the first X number of PCs that explain up to 50% of the variance, or 90% of the variance. But it becomes very clear as you look at your percent variance explained after which PC it just kind of drops off. And that's typically where you want to draw that line. So clustering, we'd like to find our groups of cells within this unknown heterogeneous group. So to define it, we would like to assign these objects to homogeneous groups. So these cells or objects we want to assign to groups that they are more similar within each other than they are and more dissimilar to things outside of this group. So for example, we have all people, a particular person, cut here, cut here. Maybe here we have, for this particular person, just their colleagues, friends, schoolmates, family members, etc., where they're not as closely tied to other groups and all people. So we describe each object by features. In this case, we have genes to explain our objects. And one important step and consideration in clustering is what is your distance metric? Typically, what we do is use Euclidean distance, often on a reduced space already. So you can Euclidean distance already on physical components. Or another very nice distance is the cosine distance, which is the angle between two vectors rather than the Euclidean distance. And one thing that can happen is the cells are clustering like you expect they should, and the distance metric can be suspect in that regard, or what you are clustering on, if that's the original genes, the entire gene set, or reduced gene set to just the highly expressed or highly variable genes, or another reduced dimension. But I have seen people go into like scikit-learn and try like all 30 or possible distances that are there, and that is, I think, a little overkill. I think pick a couple, and then if really no distance metric is working, then you probably want to go back upstream to understand your data a little bit better. So let's talk a little bit about hierarchical clustering, which can be defined as either bottom-up or top-down. So from bottom up, we start with each group is in its own cluster by itself. Each object is by itself. And we then pairwise group the objects that are most close to one another until we have one cluster at the end. Top down is exactly the opposite. Is exactly the opposite, where you have a big cluster at the beginning, split the cluster into two and keep splitting until each cell or each group or sample is by itself. Often, this type of clustering is used on the clusters themselves. So after you've done some kind of clustering on your cells, what can be helpful to see is clustering of your clusters, which sounds super meta, but it does help to understand the structure of your data a little bit more. One algorithm that you may have heard of for clustering is K-means algorithms. I personally don't recommend it for single-cell RNA sequencing, RNA-seq data. I'll explain a bit why, but I think it's a fundamental algorithm that should be in your minds at least. So given some data, so these gray rectangles are the data here, and we pick, so in this case we have k is equal to 3, so 3 initial means of our data, and these are randomly chosen. And from each data, you create those k clusters, so for each data point that's closest to, you take whichever mean is closest to it, and assign all of those to a cluster. So this one rectangle got assigned to the red cluster, all of these got assigned to the green cluster, all of these to the blue. From there, you update your mean by taking this original mean and then looking at the new data, and where is this new mean? And you continue until you converge to the number of clusters where your means don't update anymore. The reason why this can be troublesome for single-cell RNA-seq analyses is because if you have one cluster that you know exists and is very small, another cluster that you know exists and is very large, because you're randomly initializing your mean, you will by chance more likely initialize in the larger cluster. So the signal from the larger cluster will somewhat overwhelm, and it can be really hard to pick up those smaller clusters. So K-means works really well if all your clusters are the same size, about the same number of samples. But if they have different number of samples, it can be more difficult to pull out the larger and smaller clusters. Now, let's talk about graph-based clustering. I would say this is now the standard of clustering for single-cell RNA-seq data sets is to use community graphs. So given many different people, so scientific community and then many people within, family, many people within, we're looking for communities. So this is groups, for example, in the scientists, groups that are more connected to one another than they are to others. They're still connected. There's still like a couple edges, like someone, this physicist is married to a mathematician who collaborates with biologists. Like there's still connections, but overall, we're looking for communities that are more tightly, a more tight connection within themselves than across themselves. Now, how do we find these communities? A very commonly used algorithm is called Louvain clustering. So, given a first pass of the data, so we have many clusters, Louvain tries to maximize modularity of the data. So what does that mean? That means that if you take out this particular group, that you severed the fewest number of connections. So if you take out 0 relative to 2, you have some 0.23 change, similar with 0 and 4 and 0 and 5, but 0 and 3 don't seem to be as strongly connected to other things similarly as 0 and 4, 0, 4, 0, 2, and 0, 5 are. So what that's showing is that 0 and 3 are likely not in the same community, because if you remove that connection, then you're not affecting the other connectedness as much. So in this initial step, we've assigned initial groupings of our each node, or each node here, in this case, we can think of as a cell. And now we have a little summarized version of our graph initially. So now we can say, okay, within this purple cluster, we had 14 total connections. With orange cluster, 4 total connections, or each edge is cut twice each direction, and so on. Now we can take a second pass on that data and say, okay, for this group that's 14 self edges and 4 self edges here, these are actually quite well connected within across themselves too, just as well as this one is connected to itself. So again, we can call these the same cluster and merge them. Now, this Louvain clustering was named after a town in Belgium, and recently there was a paper that coined a new, or basically a pruning phase on the Louvain clustering called Leiden. So just 2-hour drive from one to the next. And what this does is that given our community that we have initially, this is an additional pruning step at the end of your clustering to ensure that the communities that you do have are well connected. So that may mean that some clusters do need to be merged or some need to be split up, because while Louvain focuses on modularity or like removal, it doesn't focus as much on connectedness. And if you're really interested in the math behind this, I definitely suggest checking out this paper. Now, how can we visualize this data? Very often the algorithm used is called T-distributed stochastic neighbor embedding, or TSNE. So this combines dimensionality reduction, such as BCA, with random walks on your nearest neighbor network. So each cell connects to every other cell at some threshold, and from there, that's how this two-dimensional representation is found. TSNE is very good at maintaining local structure. So, as in, like I have one cell, my 5 closest friends, 25 closest friends are these cells. Let's all be together in the same place on the 2D plot. It's not so good at understanding, like, oh, I'm this cell, I really hate that other cell, I want to be as far away from this other one. It's so it's really good at getting that local structure, but not so much at the larger structure. So if you do see if there is a true continuum in your data, it may be destroyed by TSNE because it maybe you want to find that one group of cells over here, another group of cells over here, but your intermediate is showing up not exactly where you think it is, or where it would make sense in a 2D plot, but TC doesn't think about the global structure that way. So TC is stochastic, so the x and y axes don't have physical meaning. They can be really anything. TC also is really designed to make like a nice big circle, because that maximizes or minimizes the distances between all the points, like a circle. That's why a cell is a circle instead of a square. And one value that you may be playing around with TC is called perplexity. This lets you adjust the number of neighbors that you see for each cell. So if you're trying to find a really rare population, you may need to reduce the perplexity so that you only see a few cells in each neighborhood for each cell. Or if you're trying to find a larger structure, you may want to use a larger number, which will clump more cells together at once. All right, more visualization. So I mentioned that TC is great at preserving local structure and not so strong at the larger structure. UMAP is good at both the local and high-dimensional long-term structure. So given the manifold of data you originally are on, it preserves both the local structure of the original manifold and the term I'm looking for, long-dimensional structure. Yeah, longer distance structures. And so UMAP stands for Uniform Manifold Approximation and Projection. I will say that because it is more network-based, it doesn't make like the nice big circle TCs that people like to see, but it is more true to the underlying structure of the data. The other advantage is it can handle very, very large data sets, hundreds of thousands of cells very easily. It does a dimensional reduction in addition, and by preserving the local structure and the global structure, you're able to interpret the shape a little bit more readily, rather than TC where the shape really doesn't mean anything. Right, for temporal data sets, some of you may be interested in looking at temporal developmental data sets. I really love this figure from the zebrafish embryogenesis paper. And there are quite a few methods for, given your many different cells from single-cell RNA-seq, to find a trajectory within them. And I'm actually going to focus on a newer method that's called Velocitope, or RNA velocity. Basically, this is a really, really beautiful, I think, representation of what single-cell RNA-seq is really for. Like, this really uses single-cell data to its max. So the basic idea is that when you have unspliced mRNA, that is an indication that this molecule was recently transcribed. And spliced mRNA means that it's already been through mRNA processing and is less recently transcribed. This more and less recently, this is on the order of hours. So again, if you have like days and months between data sets, maybe not so applicable, but for a developmental or response data set, it could be really useful. So you can tell the difference between genes that have not a lot of pre-mRNA and a lot of mRNA. Those are ones that you can say are like on their way out. So this gives you an idea of like where the cell is going. The cell is now seems to be making less of this molecule compared to where it was in the past. So in the past, this mRNA gives you a glimpse into the cell's past. Or you can have equal amounts of pre-mRNA and mRNA, or this pre-mRNA pool is going up faster than the mRNA. That suggests that this cell is transcribing this gene a lot more quickly and a lot more recently. So this allows you, Velocitope allows you to transform each individual data point into a vector and then give you a sense of where this cell is going based on its pre-mRNA. This allows you to make really beautiful plots like this. And this is used to great success in the mouse hippocampus developmental trajectory. And So given this flow, what they saw was that here is the original cell population, and without having to know how many final cell types they have, what is the trajectory through which cell types they go through, this is a very nice, unbiased way to let the data show you what the trajectory is. Now, for briefly, for differential gene expression, this stays connected. Stay connected. No, okay. Where did my computer decide? I think my computer decided. Wow, that was exciting. Okay. While this charges. Okay, sorry about that.
I have a question that you talked about.
Yeah, let's hear it.
Can you explain more what you meant by the global structure versus the local structure?
Yeah. Sorry, can you see what it? Just don't see it. Okay, thanks. Yeah, sorry, can you ask the question?
The difference.
Okay, yeah. The local structure would be, so let's say you did have a temporal data set like that, the one that was shown there. If you are looking for a local structure, what something like TC could do is basically make little blobs of your entire data set. So the group of cells over here, group of cells over here are not necessarily connected by their actual trajectory. Like TC is such that it's a stochastic one, and it's such that like if I have my group of cells that I really like over here, and if one of them takes like one step away, like one small step, suddenly we're not in the same group anymore, and now it's in a totally different cluster. So even though like through friends of friends, me and this other cell were connected, that TC really penalizes if it's not like really closely connected. So yeah, when you're looking for trajectories for like many different cells changing across time, and they're like very tightly organized, like following one by one, then something like UMAP or Velosto may be a nice choice.
So let me know if this is the right assumption. But so the TC would be good at like telling apart different cell groups, whereas the Velocitron would be good at if you wanted to see how a certain cell type changes over time.
Let me see if I understand. So you're saying, are you asking if TC would be good at just telling the difference between, or finding different cell groups or cell clusters, whereas Velosto would be good at finding cell types that are changing over time?
Yeah.
Yeah, that's the way I would think about it for sure. Yeah.
I think it's booting now.
Yay, booting, cool. Yeah.
So I think I understand what you're saying, that the clusters, the separation of the clusters doesn't necessarily mean that they're closest. So two clusters in a Disney puzzle don't mean that they're closely associated to each other. It's just the way it does. My understanding is that when you start with Tiffany, it's a random seeding of the first event, explaining why every time you do it, your clusters may be in a different spot. Is that correct? And is there no correlation between how close these clusters are together?
Excuse me. So the question, if I can try and repeat it back to you, is whether the between different initializations of TC, whether the physical closeness of two clusters to one another will stay the same or will be different. In general, it will be different. The clusters will largely be the same, but their position on the plot will be different.
So just because two clusters are next to each other doesn't tell you that these two clusters have cells that are the closest to each other compared to the other 5 clusters.
Yeah, unless they have like a bridge of cells between them. If they just have empty space, then they're not going to be similar. Yeah.
So is there one that would be able to show, like, it would be able to cluster it, but then also show how close related the clusters are?
So for clustering, I think the graph-based algorithm would be best, the Louvain and then the Leiden algorithm to prune. And then for visualizing the clusters, TC will show you the clustering groups. UMAP will also show you how well those groups map onto more of a continuum or reflecting the entire global structure of the data, trying to resolve it into space. Any luck? Oh no. Cool. Well, sometimes it likes sometimes it needs me to. Okay. Any other questions I can answer while I'm here? While the computer is booting? No question? Yeah.
Just sort of taking a step back. So you're doing your RNA-seq and your single cell transcriptome analysis, and one of the graphs that you showed is that you can get anywhere from, you can identify anywhere from 3,000 to 10,000 genes. But for each of your cells, they're not all going to be the same, the same set of genes. So from your perspective, what is more important, sort of trying to include as many cells as possible, even though some of those cells are not gonna have any information about that gene, or throw away some of the cells so that you can have confidence in a much smaller number of genes.
So is this from an experimental standpoint or after you've done the experiment?
After you've done the experiment, you have your set of, for each cell, single cell, your unique combination of, let's say, 10,000 genes or 8,000 genes.
Yeah, so once you have done an experiment and you have your matrix of data, I think it's best to do a pretty stringent filtering on your cells to know that filter out cells that look like they're expressing too much mRNA, it could be doublets, pull out cells that are expressing too little or maybe just some debris, and then have a really high quality set of cells that you really trust moving forward. Okay, let's see if this will work. Are you hearing that? Okay. And yay! Okay, we're back. Yay, I have my notes. Okay, everything good this time. My computer should have died at the beginning. Okay. So this biological interpretation is really what we'll be interested in. Which genes are being differentially expressed between your different cluster groups, and which pathways are being differentially utilized. There's many, I should mention that for differential expression, the single cell packages that you may use, ScanPy and Seurat, all have ways to do differential expression. So the method is there, methods are there. But I want to mention another method called single cell variational inference, or SCVI. All of the methods I talked about so far are all CPU-based algorithms, and this is a GPU-based algorithm, and they have a nice little figure here describing everything that it can do. So CVI One can be used for many different things, for clustering, visualization, normalization, imputation, batch correction, and so on. How does it do it? It uses a neural network to learn both the underlying gene expression per cell, or the size factor underlying mean and standard deviation, and the latent space, meaning the underlying cell type per cell. So it learns these concurrently, and given this latent space, you can then use this latent space to do clustering and visualization and batch removal, because you have different batches, some of your latent spaces will be batches, some will be different cell types. Given these size factors, you can then impute expected counts and do differential expression from this data. And we don't have time to go into detail, I just want to show the general method. I am going to just talk about what it is. I won't be able to talk in detail about what my research is, because everything I've presented so far is like current state of the art for single-cell RNA-seq, even though like this GPU-based thing was from last year, and the field is changing super, super quickly. So I guess I'm just going to give more evidence of how it's changing quickly. So I'll talk a little bit about my own research. And I'm very interested in reference-free comparative single-cell RNA sequencing. So describing what are the similarities and differences between the same cell type across species, and can we call something the same cell type? So given cell type observed in a human, can we describe it as the same cell type found in mouse? Is it really the same cell type, or is it, and what are the differences there? Cell type found in zebrafish, if you find a blood cell in mouse, human, and zebrafish, how similar are these? Are these really the same exact kind of B cell, or are they different kinds of B cell? Are they subtly different from one species to the next? Or in even earlier organisms, such as early chordate or a tunicate, who I'm working in collaboration with people at the Hopkins Marine Station out in Monterey. And then there's many more species that we can look into. So I will just end there as a teaser, so you can come talk to me afterwards, or send me an email if you have a question. But until now, I'm just going to say thank you for your attention, and I will take any additional questions that you have.
So the BI sounded super interesting.
Yeah.
But like, when would you use that as opposed to some of the what type of experiment would you use that for?
I think when you have more discrete clusters compared to if you had a temporal data set, I think it would still be a very good. Well, I think it'd still be a very good start, even if you had a temporal data set as well. I think because it is so versatile, it does take some time to compute, so you can figure out how to use it, then set it up, have that go running, and then try some other things while that's training. I think it's a really good place to start. The authors have put a lot of thought into the algorithm to make sure it works. They're also working on another algorithm or tool that they're calling CVIN or something for annotating data. Basically given a reference data set to be able to annotate your new data off of that reference data set in the same species. Any other questions? Okay, we'll go one more time.